Welcome to the deep dive. Today we're jumping into something that's been a huge headache for pig producers. Porcine Epidemic Diarrhea Virus, PEDV. And here's the thing, it's actually a coronavirus.
Yeah.
Pretty similar, structurally at least, to the one causing TGE, transmissible gastroenteritis. So our mission today is to really unpack PEDV. Where did it come from? How on earth does it spread so effectively? What's the real impact? And crucially, what are we doing about it? Diagnosis, treatment, and maybe most importantly, prevention. Because this isn't trivial. We're talking massive economic hits. Just think, that first year of the U.S. outbreak in 2013, estimates are around 8 million piglets lost.
8 million, yeah.
Leading to, almost up to $1.8 billion in annual losses for U.S. producers alone. That's huge. So let's get into it.
Absolutely. And to really understand PEDV, you kind of have to start the basics, right? What is it? It belongs to the Coronaviridae family. Specifically, it's an alpha coronavirus.
Right.
That puts it in the same group as TGE virus, which explains why the diseases look so similar clinically.
Right, that makes sense.
But it's important not to confuse them with Porcine Delta coronavirus, PDCoV. That one causes similar signs too, but it's actually in a whole different genus.
Gotcha. Different family branch.
Exactly. And morphology-wise, under a microscope, it has this classic look. It's roughly spherical, maybe a bit variable in shape, and it's got these very distinct club-shaped spikes sticking out, the S proteins.
The corona part of coronavirus.
Precisely. Those spikes give it the crown-like appearance, but they're not just for looks. Those spikes are absolutely critical. They're the key the virus uses to unlock and infect the pig cells. And on the flip side, they're also the main thing the pig's immune system targets to create antibodies. So they're central for infection and for immunity, which makes them a major focus for vaccines.
So the weapon and the weak spot kind of rolled into one structure.
You got it. And inside that structure, you've got its genetic material, single-stranded RNA, about 28 kilobases long. It codes for 4 main structural proteins. We mentioned the spike, or S, protein that's vital for getting into cells. Then there's the membrane, or M protein, which helps assemble new virus particles. The envelope, or E protein, acts like an ion channel, also involved in assembly. And finally, the nucleocapsid, or N protein, which basically binds and protects the RNA genome. They all work together.
But that S protein is really the key player for the immune system, yeah?
It really is. It's the main target for neutralizing antibodies, the ones that actually stop the virus from infecting cells.
Yeah.
And because it's under immune pressure, it's also where we see a lot of mutations happening.
Okay, so let's talk history. This thing has quite the travelogue, doesn't it? Where did it start?
It really does. The story starts back in the UK, actually, 1971. It was first described as epidemic viral diarrhea. Initially, it seemed milder, but by 76, a tougher version, PED type 2, showed up, causing significant death in newborn piglets. The sort of reference strain from that era, CV777, was isolated in Belgium in 78.
And then it just stayed in Europe.
Well, that's the weird part. By the 1980s, it actually became pretty rare and mild in Europe, almost seemed to fade away there for a while. But then it moves to Asia, early 80s, China, Japan, South Korea. And there it didn't fade away. It became endemic and much, much nastier. Especially after 2010, these highly pathogenic variants popped up in China, causing just devastating mortality in piglets, close to 100%.
Wow. So Asia became like a training ground for it to get worse.
You could sort of think of it that way, yeah. An evolutionary crucible, honing its virulence.
And then North America. We thought we were safe.
Yeah.
Considered a foreign animal disease.
For decades, yes. Until May 2013, it just suddenly appeared, Indiana.
And it hit hard because our pigs had basically zero immunity.
Exactly. A completely immunologically naive population met this highly pathogenic Asian strain. It was catastrophic.
The speed was just incredible, wasn't it? Over 200 herds, 13 states in just 2 months.
Unprecedented. And within a year, 27 states. That first year, as you said, 8 million piglets dead. Losses up to maybe 1.8 billion. Just staggering.
And the source? It seems almost too simple, but the USDA pointed to feed totes.
Yeah, contaminated flexible intermediate bulk containers. Those big bags used for feed. The USDA investigation concluded it was the simplest and most plausible explanation for how it got in and spread so fast across the country.
Just goes to show how interconnected everything is. A contaminated bag potentially sparking a billion-dollar crisis.
Absolutely. And that U.S. pandemic wasn't the end of the story globally. It kind of kicked off a resurgence. We saw large outbreaks pop up again in South Korea, Japan, Taiwan, even back in Europe.
So it's still very much a global threat, dynamic, persistent.
Definitely. It keeps exploiting vulnerabilities in trade and biosecurity, which brings us to how it actually spreads.
Right, the main way is still fecal oral, I assume. Pig eats contaminated manure.
That's the primary route, yes. Susceptible pigs ingest it. And infected pigs, especially those little pre-weaned ones, shed absolutely massive amounts of virus.
And it doesn't take much virus to cause infection.
Remarkably little. The infectious dose is very low, makes it incredibly easy to transmit.
Okay, so direct contact is one thing.
Mm-hmm.
But what about indirect routes? The more surprising ways it gets around.
Oh, there are plenty. Fomites in animate objects are huge. Transport trucks are a major headache.
Yeah, I've heard about that. Trailers are hard to clean perfectly.
Extremely hard. Studies show standard cleaning might not be enough. And get this, truck cabins can be just as contaminated as the trailer itself.
Seriously, wow.
And there's contaminated feed. We know the virus can survive for weeks in feed, especially wet feed. Maybe a week in dry feed at room temp? That's a proven risk.
And people, equipment, moving between barns or farms.
Classic fomites. Easy way to carry the virus if you're not careful.
But weren't there even more surprising discoveries? Like airborne spread.
Yeah, that was a big one. Evidence suggests aerosolized virus particles can travel maybe up to 10 miles from an infected farm.
10 miles. Through the air.
Potentially, yes. It helps explain some of those outbreaks where farms get infected with no obvious direct links.
That's slightly terrifying. What else?
Well, more recently, in 2021, there was direct evidence for vertical transmission, finding PEDV RNA in the testes and umbilical cords of newborn piglets from infected sows.
Meaning they could get infected in the womb? Before they're even born?
It suggests that possibility, yes, in utero infection. And then there's also transmission through contaminated milk from a shedding sow directly to her piglets while they're nursing.
Man, this virus is sneaky. It uses every possible avenue.
It really does. It's a master infiltrator, which is exactly why biosecurity has to be so incredibly tight, covering all these potential pathways.
And does this adaptability mean we're dealing with different versions, different strains of the virus?
Yes, absolutely. And that genetic diversity really matters for how severe the disease is. Broadly, we talk about genogroup 1, G1, and genogroup 2, G2. That original European strain, CV777, that's G1. The really nasty ones from Asia and the US pandemic, mostly G2.
Yeah.
A more practical way vets sometimes talk about it is S. indolel versus non-S. indolel strains. It relates to specific genetic changes in the spike protein.
And one is worse than the other?
Generally, yes.
Yeah.
The G1 or SE ogonel strains tend to cause milder disease, but the G2 non-SE indolel strains, those are the highly pathogenic ones. That's where you see the 80 to 100% mortality in naive baby piglets. Just devastating.
Right. Walking into a farrowing house and seeing that, unimaginable.
And what keeps it challenging is that PEDV mutates quite rapidly for an RNA virus. Its mutation rate is pretty high.
What's always changing?
It's constantly evolving, yes. And it can also undergo genetic recombination, mixing bits of its genome with other coronaviruses. This leads to new variants popping up.
Which complicates things like vaccine effectiveness, I bet.
Exactly. It's this ongoing arms race between the virus evolving and our efforts to control it. That S protein is right at the center of it all.
So, okay, the virus gets into a herd. How fast does it start causing problems? What's happening inside the pig?
It happens fast. The incubation period is usually just 1 to 4 days.
That quick.
Yep. Once a pig swallows the virus, it heads straight for the small intestine, specifically the jejunum and ileum. Its targets are the mature enterocytes. Those are the cells lining the villi that are responsible for absorbing nutrients.
And it just destroys them.
It infects and destroys them, yes. This leads to the key pathology. Severe villus atrophy.
Villus atrophy. So the lining of the gut gets damaged.
Severely. Think of the villi as tiny fingers lining the gut to increase surface area for absorption. PEDV infection makes them shrink, become blunted, sometimes almost disappear.
Okay, so less surface area means the pig can't absorb nutrients or water properly.
Exactly. Profound malabsorption. The gut can't do its job, and worse, fluid gets pulled into the gut lumen. Huge amounts of fluid.
Leading to that characteristic diarrhea.
Profuse, watery diarrhea. That's the hallmark sign.
And for the little piglets, that fluid loss is what's lethal, not the virus itself directly.
Precisely. They die from the consequences. Severe dehydration, electrolyte imbalances, metabolic acidosis. Their bodies just can't cope with losing that much fluid so quickly.
Why are they so much more vulnerable than older pigs?
It comes down to regeneration. Their gut lining, those enterocytes, regenerate much more slowly than in older pigs. They simply can't repair the damage fast enough to keep up with the fluid loss.
That makes sense. Explains the age difference in severity. So let's talk clinical signs. For those newborn piglets, less than a week or two old, what does it look like?
It's acute, it's dramatic. You see sudden onset of really watery, sometimes yellowish diarrhea. Vomiting is common too. They dehydrate incredibly fast, become lethargic, weak. You'll see sunken eyes.
And the survival rate.
In a naive herd hitting those young piglets, morbidity, the number sick, approaches 100%. And mortality, 80 to 100%. It can wipe out entire litters.
Just awful. And on necropsy, what do you see?
The intestines, especially the small intestine, look really thin-walled, almost translucent, and they're typically filled with yellowish, watery fluid.
Okay. What about slightly older pigs? Nursery, finishers.
They get sick too. Similar signs, acute watery diarrhea, vomiting, they go off feed. But for them, it's usually self-limiting.
Meaning they recover on their own?
Mostly, yes. Mortality is low in older pigs. The big impact there is economic day. They don't grow well, feed efficiency tanks. So it still costs producers a lot, even if the pigs survive.
Right, lost performance. And the adults, sows and boars.
They usually have the mildest course, maybe some transient diarrhea, maybe go off feed for a bit, seem a bit lethargic. Morbidity can vary, maybe 15 to 90% might show signs, but mortality is very rare in adults.
But the main impact for them is losing their piglets, right? And maybe reproductive issues.
Exactly. The loss of litters is the devastating part for the breeding herd. And yes, it can disrupt reproductive cycles too, another economic hit.
Given that PEDV, TGEV, and PDCO look so alike clinically, you can't just diagnose it by looking, can you?
Absolutely not. They are clinically indistinguishable. Same signs, same gut lesions. You have to use laboratory diagnostics for a definitive answer.
So what samples do you need?
For live pigs, ideally you want fresh feces or rectal swabs from several animals right when they start showing signs, like within the first 24 hours of diarrhea.
Right.
Oral fluids are also really useful, especially for monitoring the whole herd. You hang ropes, the pigs chew on them, and you test the saliva collected on the rope.
Clever.
And environmental swabs can tell you if the virus is lingering in the barn or how well cleaning worked.
What if the pig has already died?
Then postmortem samples are key. You need intestinal tissues, jejunum, ileum collected immediately after death. You take some fresh for molecular tests and some fixed in formalin for looking at the tissue structure, the histopathology. Timing is critical to avoid autolysis, the tissue breaking down.
Right, so samples get to the lab. What tests do they run?
The workhorse is molecular detection looking for the virus's RNA. Real-time RT-PCR is the most common.
PCR.
Okay, it's super sensitive, pretty fast, and tells you if the virus is actively being shed. The result often comes as a seat value.
Seat value.
Cycle threshold. Basically, a lower seat value means there's more viral RNA in the sample. So a low seat in a pig with diarrhea is strong evidence of active infection.
Gotcha. More virus equals lower number.
Exactly. Then you can also look for viral protein, the antigen. Immunohistochemistry, or IHC, is done on those fixed tissues. IHC is great because it visually confirms the virus is right there in the damaged parts of the gut. It links the virus directly to the lesions.
Proves causation, essentially.
Right. There are also rapid antigen tests, like ELISAs. You can potentially use Penside, but they're generally less sensitive than PCR.
What about antibody tests? Serology.
Serology looks for the pig's immune response, the antibodies. ELISAs can detect antibodies in blood serum, milk, colostrum, even those oral fluids. But antibody tests tell you about past exposure or vaccine response, not necessarily active acute disease. Useful for herd monitoring, but not for diagnosing the immediate cause of diarrhea.
Okay.
And the gold standard for measuring protective immunity is virus neutralization, or fluorescent-focused neutralization assays. But they're more complex, more expensive, usually done in specialized labs.
So with PDV, TGEV, and PDKV being identical clinically, how do labs tell them apart reliably?
That's where multiplex PCR comes in. It's absolutely critical.
Multiplex mean?
Meaning the single PCR test is designed to detect all three viruses, PDV, TGEV, and PDKV, simultaneously from the same sample.
Okay. One test, three answers.
Exactly. That's the standard of care now for differential diagnosis. You need to know precisely which coronavirus or maybe even combination of viruses you're dealing with.
Okay, so the lab confirms PDDV. It's an emergency. What's the immediate plan? You mentioned no specific antiviral drug.
Correct. No magic bullet. So immediate management is all about supportive care. Fight the dehydration.
Fluids, electrolytes.
Yes, oral or even injectable fluids and electrolytes for severely affected piglets. Providing supplemental heat is crucial for weak piglets. They get cold easily.
Makes sense.
And while antibiotics don't kill the virus, they're often used to prevent or treat secondary bacterial infections that can kick pigs when they're down, especially with that damaged gut.
And getting colostrum into newborns is vital too.
Absolutely critical. That first milk is their only source of initial passive immunity.
Now, I've heard about a strategy called feedback. It sounds intense. What is that?
It is intense, and it's somewhat controversial, but widely used during outbreaks. It's essentially controlled exposure. You intentionally expose the entire breeding herd sows and gilts to infectious material, usually ground up intestines or feces from piglets that died from PEDV.
You feed it back to the sows.
Yes. The goal is to get all the sows infected quickly and simultaneously. This stimulates a strong, uniform immune response across the herd.
And the point is to boost immunity for future litters.
Exactly. It specifically aims to stimulate that gut memory CGA axis we talked about. The idea is that by exposing the gut, you trigger the sows to produce high levels of specific IgA antibodies in their milk.
So the next batch of piglets gets protection through the milk.
That's the strategy. It can help shorten the duration of the outbreak in the farrowing houses by providing better passive immunity sooner. But it's not without risks.
Like spreading other diseases.
That's the main concern. You don't always know what else might be in that feedback material besides PEDV. Strict protocols are needed.
And reporting is mandatory now too, right, since the big pandemic?
Yes. In the U.S., PEDV became federally reportable in 2014. Positive cases have to be reported to animal health officials. And infected herds need a herd management plan focused on preventing spread things like tracking movements of pigs, vehicles, equipment.
Okay, so that's immediate outbreak response.
Mm-hmm.
What about the long game, preventing it from happening again? You mentioned protecting piglets relies heavily on the sow's immunity. Why are they born so defenseless?
It's fascinating biology. Piglets are born essentially a gamma globulin-anemic.
Meaning no antibodies.
Pretty much none circulating in their blood. Unlike humans, where antibodies cross the placenta, the pig placenta, it's called epitheliochorial, doesn't allow maternal antibodies, like IgG, to pass through.
So they pop out with no immune protection from mom.
Correct. Their entire initial defense comes passively after birth from drinking the sow's colostrum and milk.
Colostrum first.
Right, that first milk is packed with IgG, which gets absorbed and provides systemic protection throughout the body.
But for a gut virus like PEDV, you need protection in the gut, right?
Exactly. And that's where the regular milk comes in. It's rich in secretory IgA, or SIGA. The SIGA isn't absorbed systemically. It stays in the gut lumen and directly neutralizes pathogens like PEDV right where they attack.
Okay, so how does the sow know to put PEB-specific antibodies in her milk?
That's the beauty of the gut mammary CGI axis. It's a specialized immune pathway. When the sow is exposed to a pathogen in her gut, maybe through natural infection or feedback, or an effective oral vaccine, specialized immune cells in her gut get programmed to make IgA against that specific pathogen. Those programmed cells then travel, or home, to the mammary gland, and there they start churning out large amounts of that specific CGA, which gets secreted directly into the milk.
Oh. So the milk is basically customized immunity based on what the sow recently encountered in her environment.
Precisely. It ensures her piglets get protection tailored to the threats they are likely to face immediately after birth. It's a brilliant system.
And the whole point of sow vaccination for PEDV is to trigger that system effectively.
That's the primary goal. Stimulate that gut memory axis to maximize protective CGA levels in the milk for the piglets.
Which sounds great, but you mentioned earlier that vaccines have challenges. It's not a perfect solution yet.
No, it's definitely not perfect. Achieving complete sterilizing immunity with PEDV vaccines has been really tough.
Why is that?
Several reasons. One is strain mismatch. Older vaccines were often based on G1A strains, but the really dangerous strains circulating now are mostly G2. The protection might not cross over perfectly.
So the vaccine might be targeting an older version of the virus.
Potentially, yeah. We often see what's called leaky protection. The vaccine reduces the severity of the disease, which is good, but it might not stop the pigs from getting infected or shedding the virus entirely.
Okay.
And a really fundamental challenge is the route of administration versus the site of protection needed.
You mean injecting it in the muscle?
Right. Most vaccines are given as intramuscular injections. This generates a good systemic IgG response, which is important. But for PEDV, we desperately need that mucosal SIGA response in the gut delivered via the milk. And IM injections just aren't the most efficient way to stimulate long-lasting, high-level SIGE production in the mammary gland.
So we're injecting here, but we need the protection there in the gut. That's the mismatch.
That's a core part of the challenge, yes.
So what kinds of vaccines are available trying to overcome this?
Well, you have the traditional types. Inactivated or killed vaccines. They're safe, can't cause disease, but they usually need adjuvants to boost the immune response and often require multiple doses, typically given IM.
Okay.
Then there are live attenuated vaccines, LAVs. These use a weakened form of the live virus. They tend to mimic natural infection better, giving a stronger, broader immune response. And importantly, some LAVs can be given orally or intranasally.
Which might be better for that gut-memory axis.
Potentially, yes. Better stimulation of mucosal immunity. The theoretical downside is a very small risk they could cause mild disease or, even more rarely, revert back to being more virulent, though modern ones are designed to be very stable.
Gotcha. Any others?
Subunit vaccines. These use just specific purified parts of the virus, like the S protein. Very safe, no risk of replication, but again, they usually need strong adjuvants to work well.
And what about newer technologies? Anything on the horizon?
Oh, yeah. There's definitely research into next-gen platforms, things like mRNA vaccines. We all heard a lot about those recently. They can be developed rapidly and tend to induce potent immune responses.
Right.
And also things like replicon vaccines or viral vector vaccines, which use a harmless virus to deliver the PEDV antigen. These can potentially give strong, sustained immunity. Lots of ongoing work there.
So for producers using current vaccines, what's the typical strategy for sows?
The focus is always on maximizing antibodies in that colostrum and milk right when piglets need it. So typically, you vaccinate pregnant sows and gilts, usually a primary series for gilts, maybe 2 doses, and then a booster for everyone before each farrowing, oftentimes something like 5 weeks and 2 weeks before they're due to give birth.
Timing it for peak antibody transfer.
Exactly. And there's increasing interest in using those mucosal routes or prime boost strategies, maybe priming with one type of vaccine and boosting with another, trying to get the best possible immunity where it counts in the milk.
But even with the best vaccine program, it sounds like biosecurity is still king.
Absolutely. Non-negotiable. It is the single most critical component for long-term control and prevention.
And it's more than just fences and disinfectant, right? It's about people.
It's fundamentally about managing human behavior. You can have the best protocols written down, but if people don't follow them consistently, day in, day out, the system fails. Outbreaks often happen because of failures in compliance, not necessarily flaws in the plan itself.
So what's the core idea behind good biosecurity for PEDV?
The central principle is the line of separation, the LOS.
Line of separation.
It's a clearly marked boundary. Think of it as dividing the world into the clean inside the farm premises, the barns, and the dirty outside. And the rule is, nothing crosses that line inwards without undergoing a specific, validated cleaning and disinfection or entry protocol. People, animals, vehicles, supplies, everything.
Keeping the outside out.
Precisely. Preventing the virus from getting onto the farm in the first place. And if it somehow does get into one part, containing it there.
So what are the critical points where that line needs to be defended?
Oh, several key areas. Personnel is a big one. Strict rules about downtime, meaning how long someone has to be away from other pigs before entering your farm, could be 48, 72 hours or more.
Okay.
And controlled entry points. The Danish entry system is common. You have a bench or barrier, take off dirty outside closed boots on one side, swing your legs over without touching the dirty side, put on clean farm-specific closed boots on the other.
Creates a physical break.
Exactly. Shower in, shower out is considered the gold standard, especially for high-health herds.
What about trucks? Those seem like a major risk.
Huge risk, especially swine transport trailers. Mandatory cleaning and disinfection protocols are essential. The TADD protocol is often cited. Thorough wash using all-over detergent, then proper disinfection, and critically allowing time for drying. A wet surface compromises disinfectant.
Drying is key.
Very key. And establishing a stripped line of separation at loading docks so the truck driver never actually enters the clean barn area. Farm staff handle pigs on the clean side only.
Makes sense. Bringing new animals in.
Always risky. Best practice is sourcing from herds known to be PEDV negative. Then, all new animals or animals returning from shows or other sites must be quarantined.
Isolated.
Yes, in a separate facility away from the main herd for a minimum of 30 days, ideally longer, with testing done during quarantine before they're allowed to mix. And strict workflows handle quarantined animals last in the day, shower, or change completely before going back to the main herd.
What about feed and supplies? We know feed can carry it.
Absolutely. All incoming supplies need a decontamination step. Maybe passing through a UV chamber or being wiped down with disinfectant at a dedicated entry point or pass-through window, never just brought straight into the clean area.
Yeah.
And don't forget pest control. Rodents, birds, even flies can potentially move the virus around. So robust pest management is part of biosecurity.
And dead animals. Carcass disposal.
Needs to be prompt and done in a way that prevents cross-contamination. Ideally off-site removal using dedicated equipment that doesn't re-enter clean areas without thorough cleaning.
Wow. It's comprehensive. It touches everything.
It has to be, because the virus can exploit any gap.
And keeping it all working requires constant effort.
Constant vigilance. Regular auditing, checking that protocols are actually being followed correctly, and ongoing training for everyone, staff, contractors, truck drivers, anyone who comes near that line of separation.
Because biosecurity is only as strong as.
The people who implement it. Every single person plays a role. It's not just a set of rules, it's a culture you build on the farm.
So wrapping this up for everyone listening, this deep dive into PEDV, it really highlights maybe 3 key things for control.
I think so. First, you need rapid, accurate diagnosis, knowing what you're dealing with, especially using those multiplex PCR tests to differentiate it from TGEV and PDCO. That's step 1.
Diagnosis.
Second is smart herd immunization, particularly focusing on the sows, really leveraging that gut mammary CIGI axis to get protective antibodies into the milk for the piglets.
Immunization.
And third, the absolute bedrock. Unyielding, comprehensive biosecurity, managing every possible entry point, and critically ensuring consistent human compliance.
Biosecurity.
Yeah.
Diagnosis, immunization, biosecurity. The 3 pillars.
Pretty much sums up the core strategy, yeah.
You know, looking at the bigger picture, this whole struggle with PEDV really shows how interconnected our global food system is, doesn't it?
It absolutely does. And it's a stark reminder of this constant dance between pathogens evolving and our efforts to stay one step ahead. They adapt, we adapt.
So maybe a final thought for you, the listener, to chew on. What lessons can we take from this ongoing battle against PEDV? What does it teach us about the need for vigilance, for adaptability, for these really comprehensive systems-based approaches when we face other major health threats, not just in animals, but potentially in human health, too, in this world where new challenges seem to emerge constantly? Something to think about.